Thank you so much. And thank you again for the invitation. And it's my pleasure to be here to tell you a little bit about our past work and then kind of lead into what was the motivation behind developing this mosaic microscope. I'm going to tell you about that towards the end of my talk. As is, I want to just start off by actually acknowledging the most important part of my journey is my collaborations with a host of amazing people from Harvard, where I did my postdoctoral work, Eric Betz's group at Janelia when he was still there, his group. The Megasin Lab, most of the zebrafish work that I'm going to show you was in collaboration with the Megasin Lab. The Drubin Lab is now my colleague upstairs who develops like gene edited organoids. I'm going to show you some examples using adaptive optical lattice light sheet. And then we worked with Ed Boyden's group on combining lattice light sheet microscope with expansion microscopy, so you can get scalable super resolution on a tissue scale. Let's get started. So really the only introduction you need for the next few minutes is that, okay, well, you can take this beautiful cell on the cover slip. If you want to image the ventral dynamics that are attached on the surface, you don't need to use anything other than turf because that confines the excitation almost entirely within the depth of focus of this objective. It gives you amazing contrast and great signal to noise. Really, there's not a better tool for studying these ventral dynamics. However, if you want to study anything away from the cover slip, you need to do some sort of confocal sectioning to be able to get a good signal to noise. And the way that happens is using a pinhole to focus the light at the particular depth of focus. And you can change the depth of focus either by moving the objective or by the sample. But you notice that you're illuminating the entire volume of the cell for collecting a single point over here. Of course, you can multiplex this in a spinning disk context where you're collecting 2D optical sheets by using a bunch of pinholes focusing the light. But again, notice that for every 2D optical section, you're basically illuminating the entire volume of the cell. So it's highly photon inefficient. Light sheet technology has been around for about 2 decades, and it's an amazing and transformative tools, especially when it started out in the application space of being used for developmental biology. It's great because you're able to get cellular level resolution, no problem, large fields of view. The issue to study subcellular dynamics becomes that using traditional diffracting light sheets, such as like a Gaussian light sheet, tends to give you relatively thicker light sheets. Thicker meaning that it's either larger than the depth of focus of a high NA objective, or you can make it a higher NA light sheet, but then it kind of compromises your field of view because it's converging and diverging very rapidly. And this was until Eric Betz's group in 2014 developed the lattice light sheet, where they used non-diffracting beams, basically placing Bessel beams at particular periods. So if you take a cross-section of the light sheet, you have these Bessel beams. And a single Bessel beam by itself is still a pretty poor choice for a light sheet because you have the energy in these higher orders. But when you bring these Bessel beams at particular periods or particular lattice positions, what you're able to do is interfere out these higher orders such that you get basically most of your energy confined within the central order. Of course, this still doesn't look like a light sheet. So what we do is we use a galvanometer to spin it or to sweep it faster than our exposure time to create a very thin propagating light sheet. Of course, we can take advantage of the modulation in signal and phase and use it for structural illumination microscopy for 3D super resolution live imaging. So what does this all mean practically? Well, let's take these gene edited cells. So these gene edited cells have been gene edited for or fused with a GFP on a adapter protein that's responsible for generating these self-assembling cloth-encoded pits. Basically, cloth-encoded pits are responsible for how cells eat or bring in majority of the nutrients into the cells. So if you're now looking at these gene-edited cells, what we do is we've imaged using the spinning disk confocal and the lattice light sheet, and we're imaging it just as fast in the lattice as the spinning disk, this particular spinning disk can handle. And we put in just enough power such that the starting signal to noise for both of these volumes is about the same. And what you're looking at here is the top-down view. So this is like a volumetric sample. You'll see basically that about almost 50 times faster photobleaching on a spinning disk confocal if you're doing 3-dimensional volumetric imaging compared to the lattice light sheet. So with this, we kind of apply this technology to a host of different problems over the years. So we've looked at, how phosphodienstatide conversion takes place at the site of endocytic vesicles. We've looked at how viruses start to internalize into these cells. In this particular case, we're looking at how rotavirus particles, so these are pseudoparticles that have been reconstituted with different inner core material and outer core material, such that when you lose the outer core material, which is the green, that's basically when it has access to the sialic acids in the plasma membrane to be able to internalize itself. We've looked at S-core dynamics. So this is now trying to understand mechanistically the dynamics of how you form these AAA ATPase polymers in order to create interluminal vesicles. We're starting to look more closely at how the trafficking is taking place between ER and Golgi, and this is like active work that's taking place. And then all of the examples I've shown so far are of cells on a piece of glass. What we wanted to do is be able to achieve the same level of resolution, diffraction limited resolution that we have in these settings, but be able to do volumetric imaging inside of living or physiological systems like organoids or intact transparent animals like zebrafish. When you don't care about life in certain instances, we want to be able to get more resolution out of them. That's the story I'll briefly tell you about. Okay, well, what happens when we combine expansion microscopy, which is a way to kind of bypass the laws of physics to be able to attain super resolution with this method. So just as, again, frame of reference, this is a single cell. I'm just kind of rotating that cell around so kind of get a visual for the three-dimensional nature of this particular cell. Using the lattice light sheet, this gene edited cell, we're kind of now being able to track every single endocytic event that's taking place in the entire volume of this particular cell. Well, this is great because we get to learn a lot, especially when we repeat this several dozens of times in dozens of cells. We can kind of characterize the different behaviors of each of these endocytic pits and the diversity of behaviors that these pits in the entire volume of the cell present. This is great, especially because we're looking at this in a simplified system. But the question remains, how can we make this more physiological? So this is work that came out a few years ago. And we started by using genome-edited intestinal organoids. So in this case, we're expressing clathrin and dynamin, and these are embedded and kind of held together in these matrigels. As you can see, right off the bat, you basically don't have these diffraction limited spots anymore. You basically have more like a fuzz. The reason we have a fuzz is because the light sheet that's traveling to the region of interest in this particular sample or its organoid is basically hitting the cytosol and the nucleus, of various cells. It's passing through. Every time you kind of go through these changes in refractive index, the light sheet starts to scramble. So by the time your light sheet kind of gets to where you're trying to image, it's already kind of completely scrambled. Anything you do manage to excite here is going to try to make its way back to this detection objective, and that's light is also passing through different levels of cells and cytosol, nucleus, and other cellular material, and basically that's also getting scrambled. And the third thing is there's going to be a focus offset because of Snell's law. When this light sheet kind of hits this curved surface, it's going to bend a little bit. That means that where it focuses is no longer going to be where the two objectives are supposed to be. So we need to fix these three problems in order to be able to recover the resolution lost in these organoid systems. So an ideal cross-sectional light sheet kind of looks something like this for a multi-vessel pattern. When it kind of goes through an aberrating media, in this case, we kind of pass it through about, I think, 2% agarose, and the light sheet starts to bloom a little bit. And then what we do is we take basically the phase error of this, subtract that from the ideal transform of this, inverse transform that such that the pattern that we inverse transform, when it passes through the aberrating media, will now yield back the corrected cross-sectional light sheet. So that's basically how we fix the excitation side of things. The detection side of things, we're using a approach that astronomers have developed many decades ago, where they're trying to observe these stars from ground-based telescopes But the problem is that this light that's traveled, thousands of light years comes without any problems until it hits our stratosphere or atmosphere. And because of the moisture and the clouds and all of that, it kind of completely gets scrambled. So what they've done is they created an artificial star by exciting the sodium atoms in the stratosphere. They know the shape and size and the properties of this guide star that they're generating next to the real star that they want to image. They measure basically this by bouncing this off of a deformable mirror and a control loop where you're using a Shack-Hartman wavefront sensor to measure this aberrated wavefront and then adjust the shape of this mirror such that you now have a correction for this distorted wavefront. So you're now able to deblur basically the stars. We use a similar approach in our adaptive optical lattice light sheet, where the 2-photon creates our guide star. And instead of exciting sodium atoms, we're basically exciting the fluorescent molecules that are within our imaging region of interest. To correct for the focus, we basically send light through from both of the objectives and image them onto the same camera, and then see what the offset is and use a galvanometer to adjust the light sheet position such that it matches the focal plane of the detection objective. By implementing all of these three things into a single microscope, we're able to recover that lost resolution. So this is great because we were able to image organoids, but we want to be able to now move on to image in more complex systems. So one of our favorites is zebrafish, because like us, it's a vertebrate and it's mostly transparent. So we've went ahead and done the adaptive optical lattice light sheet on these zebrafish, and I'm going to show you a few examples. But just to give you a sense of scale, that's that single cell I was showing you that was kind of turning. And this is a multi-tiled volume that's been corrected with and without adaptive optics. And you can basically see the night and day difference it makes with adaptive optics versus without adaptive optics. For the next few examples, I'm going to show you on the bottom right, just a schematic of the fish, just to give you a sense for how old the fish is and where we're imaging. So in these few examples, we kind of continue the theme of imaging through the different tissues, but here imaging endocytosis in these different tissues. And what we interestingly found is that when we surveyed over a dozen different tissue types, we found the greatest amount of difference between the brain cells and the muscle cells, meaning that the muscle cells have much longer lived events compared to the endocytic events in the brain. And we kind of have an intuition as to why this is. And this has to do with the membrane tension of the cells in the muscles versus the brain. So that way, the muscle cells require, the endocytic pits in the muscle cells require actin to more or less complete the endocytic events in a reasonable amount of time. Next, we looked at organelle dynamics in zebrafish. So one of the challenges here is that, well, we're looking at several things at once. We're looking at membrane, Golgi, endoplasmic reticulum, and mitochondria. To visualize all of these at once is more of like a nightmarish scenario, because you basically have no perception of which cell belongs to what. So the first thing we did was we're able to leverage the membrane signal here to be able to delineate all of these cells that are in the volume of this particular tissue. And then what we did was we computationally separated out each cell such that you basically have a view of individual cells as one is used to studying, but with a preserved morphological context of all of these cells. So in this case, you can see these dividing cells here. You can see these amorphous, blobby looking skin cells over here. This is the hindbrain of the zebrafish. So these are the precursor neuronal columnar cells over there. Another mitotic cell there. And in a second, what I'm going to do is I'm going to turn off all of the cells except for just one, so that we can focus on the quality of data and the types of questions one can ask with these, with this level of detail that we have access to in these microscopes. So I'm going to turn off everything except for this one particular cell. So in this case, what we did was, because both the Golgi and the membrane were in the same spectral channel, we computationally delineated them, because we know that the Golgi and the membrane are not going to be spatially overlapped. So we have the mitochondria and the ER, and what I'm showing you here is the rough distribution of the organelles as a distance from the membrane. So when the cell starts off an interface, these organelles are roughly evenly distributed. And we can track the volume and surface area of this particular cell over time. And in a few seconds, you'll see that the cell starts to round up. So that means that it's kind of entering mitosis. So when it enters mitosis, the surface area drops. You can see the mitochondria is more peripheral than the other two organelles. And when the cell goes through cell division, the membranes redistribute, so the organelles redistribute, and the surface area of this particular cell recovers. Yeah, both of these daughter cells roughly have similar volumes and similar surface areas after cell division. In another example, I'm going to show you a couple of examples of cell motility in zebrafish. So what we've done is we've generated this xenograft model where we're able to inject human breast cancer cells into this zebrafish. So these breast cancer cells are in green over here. And the vasculature of the zebrafish is labeled in magenta, so the endothelial cells. And we kind of see different examples of cell migration. So in this case, this guy is squeezing through these narrow vessels, and then, as part of that squeezing through, it does kind of dilate the vessels a little bit, in this case over there. Over here, it's kind of rolling on top of these blood vessels, and you can kind of see these long ciliated looking structures, potentially making these cash bonds as they kind of roll through or roll on top of these blood vessels. And in the last example here, we kind of see this particular cancer cell trying to extravasate out of this blood vessel. So trying to metastasize. And we know that it's trying to metastasize because if you track the surface area of that particular green cell, it's starting to increase over the course of, increases by about 50% over the course of about 2 hours or so. And in the last example in this section, I'm going to show you, in one field of view. So we're here we're imaging, again, the ear, region of the ear of the zebrafish. So we're trying to study this endolymphatic duct and sac that's regulating pressure as the ear develops. But you can kind of see the immense diversity of biology that's taking place in this one field of view. So in this one field of view, we can basically look at these ruffling and dancing skin cells. You have this hindbrain on the bottom. You have a blood vessel right here that red blood cells are kind of zipping through. You have these endothelial cells right there. This guy particularly that enters into mitosis. Again, the endolymphatic duct and sac, the pressure relief valve that I was telling you about. Immune cells that are just crawling around, sensing and doing their duty. And it's an immensely kind of complex thing that you're able to collect in just one, region of interest, 11 different cell types in this particular volume. So with this adaptive optical lattice light sheet, what we were able to do is be able to record dynamics at the scale of nanometers and milliseconds to really understand their consequences over the scale of microns and minutes, and to really kind of visualize their long-term outcome at the scale of several millimeters over several days, several hours or several days. We can do quantitative in vivo cell biology in multicellular systems, whether it's organoids or in transparent organisms, with diffraction limited resolution, because we're able to recover that lost resolution due to aberrations. And with the right reagents, we can calibrate these microscopes such that we can get single molecule sensitivity using something like halo dyes or JF dyes. So I want to kind of switch gears a little bit and tell you about our other work where we combine lattice light sheet microscopy with expansion microscopy for, again, rapid, scalable, super resolution of tissues. Here we're looking at 2 neurons in the somatosensory cortex with vastly different pre and postsynaptic connections. But I'm going to kind of jump right into it in the interest of time. So take this tissue slice of a mouse brain, and we're not zooming in or anything like that. We've infiltrated this particular tissue with an expandable polymer resin. Once you digest it, everything out, and all the macromolecules of interest are anchored to our polymer network, we basically expand it by removing the salts. And as a consequence of that, what happens is we're basically physically expanding this tissue. That means that every molecule is still in the same place as it was, except now it's separated by the expansion factor in all 3 dimensions to every other molecule in this particular sample. And again, this we can use various types of soluble polymers. In this particular case, we're using sodium polyacrylate, and basically pre-expansion, post-expansion. One of the nice things of expansion is that it naturally clears the tissue of all of the scattering elements. So we don't need to worry about, doing the refractive index matching for, clear tissues. So that comes as a byproduct of expansion. So using this, what we've done was we were able to map with, again, better than diffraction limited resolution, all of the components in this tissue. So in this particular case, we're looking at one soma, where this is a Thigh1 YFP mouse line. So this particular soma is filled with YFP signal, but it nicely excludes all of the compartments within this particular soma. So we're able to segment out all of these compartments, be able to kind of quantify shapes, dimensions, and all of those interesting things, and be able to identify a subset of these as either mitochondria, which are going to be shown in magenta in a second, or lysosomes in yellow. And we're basically able to, again, characterize the shapes and morphologies of these organelles. And again, we've done that in the soma, we've done that in dendrites, and we've done that in axons. And you see that, you have larger mitochondria in the soma and the dendrites compared to the axons, because it makes sense, because axons are pretty constricted and very, very thin structures. So again, this is one field of view. So what can we do with, if you wanted to image something larger? Well, what we've done was we mapped the entire somatosensory cortex, this region of the tissue. So this is about pre-expansion. This is about 2 millimeters by about 1/4 millimeter by 1/4 millimeter. And it took us about 18,000 volumetric tiles per channel to be able to collect this particular volume and then stitch it together. But the quality of data is pretty impressive, in the sense that in this one giant volume, we were able to collect information about the pre and the post synapses associated with or without these glutaminergic neurons. And we were able to collect more than 100 times the information that exists in all of literature today, which is very, very cool. And we were able to kind of quantify, the distances for all of these, so you can get a broad distribution, or the Homer-Bassoon distances for objects that are associated with these glutaminergic neurons. So we can basically see that, okay, well, there's a tighter distribution for the ones that are associated with the glutaminergic neurons versus not. So again, this is still a portion, again, a very, very minor, tiny fraction of the whole mouse brain. But if you want to get a more systems level thing, we kind of went to like the fly brains, where we're able to image the entire fly brain. So what we did was we've imaged these fly brains that are expressing these signals in these dopaminergic neurons. And then here, what we've done is we've stained it with, for NC82, like a presynaptic marker. So that's like that speckly white thing that you're looking at. And we wanted to ask, okay, well, can we use expansion microscopy to be able to count and locate every single synaptic position in this entire fly brain? Well, the first thing we need to figure out is, do we even have sufficient resolution to be able to figure out the localization at these really dense regions? For example, the mushroom body is responsible for memory and learning. And we had access to this FIBSIM data set, which is volumetric EM data set, where at Genelia, it took them about 1 year to collect this volume, which is 1/100th of the fly brain. And they spent 10,000 human hours manually annotating the location of every single synapse in this particular FIBSIM. And they realized, okay, there's about 90,000 synapses in this particular mushroom body. And most of these are within 200 to 250 nanometers at their densest position. So again, being that we are diffraction limited divided by the expansion factor, so sub 100 nanometer, about close to like 70 nanometer resolution, we definitely have more than enough resolution to be able to localize every single synapse in this entire volume. So we went ahead and basically wrote the code to be able to determine that position. And turns out that in this particular tissue, there's about 40 million synapses, and only about half of them are dopaminergic neuron associated. So this is great, because this is a good segue into what we're trying to do here at Berkeley. So everything I've shown you was with what I'm referring to now as the current expansion lattice. So to image the fly brain, Took us about 3.5 days. With the MOSAIC, which is the next generation microscope that we're building at Berkeley, it's really about 50 times faster. So we can image that entire brain in less than 2 hours. And again, the expansion lattice has its own niche separate from the EM world because it's able to answer questions, of course, with an order of magnitude worse resolution than FIBSIM. But it's able to do those with molecular contrast, which the FIBSIM cannot do, and be able to kind of go at problems that are too large even for dozens of FIBSIM to be able to handle in a reasonable amount of time. So this is what the microscope that we used for everything I've shown you today looks like. And you can see here that this is on a 10-foot optical table. Majority of the components that went into building this microscope, these visible lasers, AOTF to modulate, spatial light modulator to shape the wavefront, 2-photon laser for the guide star, galvos, objective sample stages. It turns out that we can basically use all of these to build the adaptive optical or the expansion lattice light sheet microscope. But it turns out that we can also use a subset of these or all of these with additional components for different types of microscope, like a point scanning microscope or a bezel light sheet microscope, 3D phase or 3D sim or area scan. And then we figured, well, why not we just reuse these optical components and design a system such that we're basically able to leverage one microscope that's able to reuse all of these elements to basically have the functionality of about 7 to 10 different microscopes. And the added benefit is that because we have more things, we can basically impute everything with adaptive optics if that particular imaging mode is able to leverage that. So this is basically what we've designed and built. So this has 3 imaging stations. So you have the light sheet configuration for the samples that are over there. You have the inverted objective. Again, these are all the similar high-NA objectives. And then you have an upright station if you want to have a mouse or something that can basically use a lot of freedom below this particular station. So we built this at Berkeley. Again, amazing team here doing a lot of hard work. So we have 2 of these at Berkeley. And really the secret sauce of what makes this microscope possible is that we have these custom designed optoelectronics where basically you're able to push a button and open and close these mirrors that will change the light path. And it does that such that you transform one microscope into a different microscope such that you can image with multiple modalities onto the same sample if it can leverage that. So that being said, there's bigger problems with these microscopes, specifically in the context of this is 50 times faster than the previous gen. That means that we're able to generate a lot more data in a small fraction of time than what we were able to do before. So we built this advanced bioimaging center here such that we bring together the biologists, the imaging specialists, and the data scientists to be able to work together on problems, again, that are too immense for each of us to work on independently. And I kind of want to show you a little bit about what we do in terms of the computational space as well, because not only are we interested in generating great biological data sets, but we're also interested in understanding the biology. And to do that, we really need to build like quantitative tools. So my postdoc, Zhang Tao, has been working on improving the ability to do 3-dimensional point source detections. So he's able to basically pick up really weak signals and be able to quantify these with pretty impressive accuracy metrics than anything else that's been published before. Again, I have a team that's working on segmentation because this is, believe it or not a solved problem for us. We basically have diverse range of biological specimens with different signal to noise, different types of staining, and basically building 3D deep learning architectures to be able to do segmentations and being able to do basically quantifications of these cell states. We're also kind of going into molecular prediction, where we want to be able to study one molecule at diffraction limit and be able to understand its correlation and its relation to other related molecules that appear with or around at the time of this particular molecule. So we basically want to be able to predict, well, in this particular case, whether or not a clathrin-coated event will be successful in the sense that in a cause decision or will be an abortive. So we've been kind of exploring on this particular front. And then the last big position that we're focusing on is being able to make deep learning based sensorless adaptive optics, meaning that we have basically a pre-trained model that we're working to develop where it's able to recognize the aberration and we're able to correct it using a deformable mirror such that our corrected wavefront will allow us to basically get close to diffraction limit resolution. So again, the Advanced Bioimaging Center is really, the whole point of that is to make these advanced microscopes accessible, and it really is open to international investigators. So please feel free to get in touch if you have scientific problems that that could be helped by these tools that we're building. I'm happy to stop here and basically take any questions.